So why are we all here today, and why are we so excited about CRISPR? And the bottom line is it works, okay? We didn't wake up yesterday and decide we want to, edit the genome and do all these amazing things to try and help people. Scientists have been working on this for a long time, and the tools weren't readily available and they weren't good enough. But now CRISPR makes it so that everybody can readily do genome editing. For scientists in the lab, this is great. I can now go into any gene in the genome and functionally assess its role in a distinct cellular process or in a disease.
So it was only a matter of time before we imagined that there would be gene modification of human embryos. And indeed, it was only in about 2015 when this was first reported. And it raised this notion that we could use this technology not for the treatment of disease, but for actually engineering the genes within the cells of our children. And of course, this led to a significant amount of consternation and a call for regulation, prudent oversight, and the bringing together of international communities to actually discuss how we might use this.
Who will give consent to make permanent genetic changes in an offspring? Should it be the parents, or should it be government?
We also face questions like, what really is the human gene pool? Is that a real thing? Does it merit respect, restraint from alteration? What claim to moral status does that have, both ethically and in law?
CRISPR allows us to have agency, to make changes. How are we going to draw distinctions between changes that reverse illness and create a sort of a level playing field versus the kind of social justice issues that are raised if we're going to intervene in ways that create advantage? That's where I think people get concerned. Because if editing is going to be disproportionately used to give advantage, it will potentially exacerbate the disparities that we already see in healthcare. And that's why I'm deeply troubled